Hello students, myself Dr. Suhail Khan, Faculty Allen Career Institute, Kota. Today we will discuss about a very important chapter, that is sexual reproduction in flowering plants. It is the second chapter of NCERT of Unit 6. The weightage of this chapter in board exams comprises of approximately 5 to 10 marks every year. So you have to take this chapter in a very appropriate manner and in a sincere manner. The type of questions that have been asked in board exams from this chapter, that includes 1 mark, 3 mark as well as 5 mark questions. From this chapter, each type of questions like definitions as well as descriptive questions, diagram based questions as well as diagram with labeling, they have been asked each and every year. So be prepared and be positive regarding this chapter for your board exams. Today we will discuss about chapter 2 of NCERT, that is sexual reproduction in flowering plants. First of all, what is sexual reproduction? So we can understand by a simple definition of this sexual reproduction that it involves the formation of male and female gametes either by the same individual or by the different individuals of the opposite sex and the fusion of male and female gametes. It means here the two processes are involved, that is the formation of male gametes and female gametes by the process of reduction division, that is meiosis, and the other process, that is the fertilization, that means the fusion of male and female gametes. It means this sexual reproduction, it involves meiosis as well as fertilization. So sexual reproduction mainly includes meiosis, that is the reductional division, and the process of fusion of male and female gametes, that is known as fertilization. So sexual reproduction involves these two processes. Now here we discuss the sexual reproduction in case of flowering plants. As we all know that there are so many plants starting from lower plants, that is from thallophytes, bryophytes, pteridophytes, gymnosperms and angiosperms. But here we are considering the flowering plants. So here we discuss about the angiosperms, that is the flowering plants. Here we see the structure of the flower, because here we discuss about the flower, and flower is that main organ which consists of male and female reproductive organs where the male and female gametes are formed. So here is the simple structure of a flower that involves this stalk that is known as pedicel. Here is a bulging structure which is known as thalamus. From this thalamus, accessory organs, that is the sepals and petals are formed, as well as the essential organs, that is the stamens and carpels are formed. Now we consider that flower is one of the fascinating organ, means one of the attractive organ in case of angiosperms. Why we consider it as a fascinating organ? Because the flowers, they are the object of aesthetic, ornamental, social and religious and cultural values. So we consider it as a fascinating organ. This flower, it is considered as the main seat, or you can say the main site of sexual reproduction in case of angiosperm, because this is the flower which contains both male as well as female reproductive organs. Flowers, they are also considered as the morphological as well as embryological marvels. Here we are seeing these two words, that is morphological and embryological. So what do we mean by this morphological marvel? Morphological marvel, as we said that these flowers, they are the attractive organs. So externally when we see angiospermic plant, the most attractive part of that plant is the flower, as it is considered as the most fascinating organ. So we consider that this is one of the morphological marvel. And why we consider it as a embryological marvel? As we already said that flower composed of both male and female reproductive organs, that is androecium part and gynoecium part, which later on involves in the sexual reproduction. That's why we consider it as a embryological marvel. Now related to flower, here is one of the science that is known as floriculture. Floriculture means that it is the science of cultivation, breeding and marketing of flowers. So it is the science that deals with the flowers, its cultivation, its breeding, as well as its marketing. Now, a scientist named Goethe, he considered that flower is a modified shoot. So first of all, we have to discuss what is a shoot. Shoot is that part which involves both stem as well as a leaf part. Now we said that flower is a modified shoot. So why we consider it is a modified shoot? Here we describe this, that it is a modified shoot. As we know that flower, it has a stalk-like structure which is known as a pedicel. So that stalk-like structure is known as a pedicel. The free end of that pedicel, which is flattened or it may be dome-shaped, that is known as thalamus. As we have seen earlier in the structure of the flower, that the stalk-like structure is the pedicel, and that flattened or you can say dome-shaped structure, that is known as the thalamus part. Now this thalamus, as it is composed of nodes and internodes, it is considered as a stem. But we said that this thalamus is a modified stem. So why it is considered as a modified stem? Because this thalamus, it contains highly condensed or highly reduced form of nodes and internodes. That's why we consider it as a modified stem. Now there are 4 whorls, that is calyx, corolla, androecium and gynoecium, and all these whorls, they arises from the nodal regions of the thalamus. And we know that leaf is that part that arises from the nodes of the stem. So all these 4 floral whorls, they arise from the nodes of the thalamus. So we can consider that those 4 floral whorls, they are the leaves. But since they are not the true leaves, we consider, as they are originated from the nodal parts of the thalamus, we consider them as the modified leaves. So flower considered as a modified shoot because it contains thalamus, which is a modified stem, and 4 floral whorls, which are the modified leaves. That's why we consider that flower is a modified shoot. So I think you understand that why flower is considered as a modified shoot, because it contains both modified stem as well as modified leaves, where the modified stem is the thalamus part and modified leaves are the 4 floral whorls. Now, as we know that the thalamus, they contains the condensed form of nodes. So we have seen that there are 4 nodal regions from where these 4 floral whorls arises. First lowermost node from where the first floral whorl arises, that is the outermost whorl known as calyx. This is the outermost whorl. The individual part of this calyx is known as sepal. This is the individual part of this whorl. Second nodes give rise to another whorl or the second whorl, that is known as corolla. The individual part of this corolla is referred to as a petal. Third node give rise to male reproductive part, that is known as androecium, and the individual unit of that androecium is stamen. So androecium is a whorl and the individual part of that androecium is known as a stamen. Fourth node give rise to the innermost whorl, that is known as gynoecium or you can say pistil, and the individual part of this gynoecium is known as a carpel. So these are the all 4 floral whorls which we have already discussed that these are the modified leaves as they are arising from the nodal regions of the thalamus part, which is a modified stem. Now here we are starting the sexual reproduction, that means we are discussing the life cycle in case of angiosperms. So the sexual reproduction in case of angiosperm, it involves the particularly 3 events, that is pre-fertilization events, fertilization events and post-fertilization events. Pre-fertilization events, it includes development of pollen grains, development of ovules, and it also includes the pollination. Then second step is second event is the fertilization event, which refers to the fusion of male and female gametes. And the third event, that is the post-fertilization event, that includes the development of embryo, endosperm, formation of fruit and formation of seed. So first of all here we discuss about the pre-fertilization events. Pre-fertilization events. In case of pre-fertilization events, there are certain hormonal changes will occur or certain structural changes will occur inside the plant which leads to the development of the floral primordia. Here a term comes floral primordia or floral primordium. Whenever this term comes, that is the primordium, that primordium means the initial structure. Any part, any plant part which is just arising from that in that plant, that initial stage of that part is known as a primordium. Here we are considering about the flower, so we can say the floral primordium. If that will be the case of in case of leaf, then we can consider it as a leaf primordium. So primordium means an organ in its earliest stage of development, or you can say it is the foundation of subsequent development. Now, when the flowers are arising on the floral axis, first of all, inflorescences are developed. So inflorescences, they are formed which bear the floral buds, as well as then these floral buds are transformed into the flowers. So what are inflorescences? Inflorescence, as we have already discussed in the earlier classes, that inflorescence, these are the arrangement of clusters of the flowers on a floral axis. Now first of all, we discuss here about the male reproductive world, that is androecium. Male reproductive organ, that is known as androecium, it consists of whorl of stamens. As we have already discussed that androecium is a whorl, but the part or the individual unit of this androecium is known as a stamen. So stamen is the individual part. The number and the length of these stamens, they are variable in flowers of different species. Stamen is equivalent to microsporophylls. Now here a word comes microsporophyle. So first of all, we come to know here about the sporophylls. What are sporophylls? So sporophyll means a leaf-like structure that bears the sporangium where the spores are formed. And in case of stamen, as we already said, that is a modified leaf. So we can consider it as a leaf where the microsporangium are formed, in which the microspores are formed. That's why we considered that stamen, it is equivalent to microsporophyll. So we should know that this sporophyll is the leaf-like structure. So sporophyll means the leaf-like structure that bears the sporangia. A typical stamen is differentiated into two parts. That is a long stalk-like structure which is known as a filament, and the terminal part which is generally bilobed, it is known as anther. So if this is the stalk-like structure, then this is the filament part, and here this is the bilobed structure, this is the anther part. So this filament, this is the filament, and this is the anther. This is the anther, and this stalk-like structure is known as filament. This is the filament. And in general cases, this anther is bilobed in nature. The proximal end of the filament is attached to the thalamus or the petal. If we are considering that this is attached with this thalamus part, so this is the thalamus part, this is the thalamus part. If this is the thalamus part and this is the filament and these are the bilobed structure, that is the anther lobes. So the part of the filament which is near or attached to the thalamus part, this is the proximal part. This part is the proximal part. And the part which is attached with this anther lobe, this is the distal part or distal end. This is the distal end. So here it comes that the proximal end of the filament, it is attached to the thalamus or the petal. That is, this is the proximal end which is attached to the thalamus, and this is the distal end which is attached to the anther lobes. anther and filament, or two anther lobes that are attached together with the help of a region called connective. That means if we are talking about this filament part, it is connected to, in this region, this is known as the connective part, and these two anther lobes, they are also connected by this connective. So anther lobes, the two anther lobes, they are also connected by this connective, and this filament, this is also connected to the anther lobes by this connective. So this is the connective. This is the connective. And this connective is actually, it contains the vascular tissues. This is the stamen part containing anther and the filament, and this is the three-dimensional view of the anther that indicates the pollen sacs in which the pollen grains are formed, and when these pollen grains after maturation released, they are coming outside from those pollen sacs, and that exhibits the line of dehiscence. A typical angiosperm that we have already discussed, that is bilobed in nature, means each of the anther, it contains the two lobes, and each of the lobe contains the two theca, that is the two space. That is, if each of the anther lobe contains two theca, then that condition is said to be dithecous condition. When we take a cross-sectional view or a transverse section of that, then a longitudinal groove, it runs lengthwise, separating those two theca out. The anther is consisting of 4 microsporangia located at the corners, 2 in each lobe. Suppose if we are taking this is the two lobes of the anther, and this is each of the anther lobe contains two spaces. Here the spaces that are considering as the theca. So condition is said to be dithecous. Bilobed condition and dithecous condition is present in this stamen or this anther. The anther is consisting of 4 microsporangia. Since these theca later on converted into microsporangia where the microspores are formed. Here only the microspores are formed. Suppose these are the microspores, these are formed. So this is the structure where the spores are formed. So we consider this as a microsporangia. So in this bilobed structure of the anther, total 4 microsporangia are formed, and that condition is said to be tetrasporangiate condition. So in a typical angiospermic anther, we see that the condition is bilobed in nature, having 2 anther lobes, each of the anther lobe composed of 2 theca. So condition is said to be dithecous in nature, and as a whole in both the lobes it comprises of 4 microsporangia. So the condition is said to be tetrasporangiate. So here a typical angiospermic anther is bilobed, dithecous and tetrasporangiate in nature. This is the transverse section of anther showing 4 microsporangia and their anther walls. Suppose this is the cross section or the transverse section of the anther, and here we see these are the 4 microsporangia. This is the 1 microsporangia, this is another microsporangia, this is third, and this is the 4th microsporangia. So these are the 4 microsporangia in which the microspores are formed, which later on maturity termed as a pollen grains. So microsporangia later on termed as a pollen sac also. So the condition here is said to be tetrasporangiate condition. Now the structure of anther. In the transverse section of the anther, it seems tetragonal in nature. Here it is considering as that if we take the transverse section of the anther, it appears to be a tetragonal in nature, means four-sided. Microsporangium, which is present here in the four corners, they are generally surrounded by four different wall layers, and these four different wall layers are epidermis, endothecium, middle layers, and the tapetum. The outermost one is this is the epidermis, the next one is the endothecium, then inside the endothecium is the middle layer, and the innermost one is the tapetum layer. We will discuss now all these four different wall layers step by step. First of all, about the epidermis. Epidermis is the outermost layer. It is the single celled thick layer. As it is exclusively involved in the protection, so it is also known as the protective layer. Just next to the epidermis is the endothecium layer, which is also termed as fibrous layer. Now, endothecium, it is present just below the epidermis. It is also single celled thick, and its nature is hygroscopic. What is hygroscopic? Hygroscopic means that it has the water absorbing capacity. It have the water absorbing capacity, and this is mainly because of the presence of fibrous thickenings in its walls. Now, on which wall these fibrous thickenings are present, we will discuss it later. The main function of this endothecium layer is that it helps in the dehiscence of anther. If we are talking about the endothecium, then it composed of three walls: outer wall, inner wall, as well as the radial wall. The outer wall, it is thin, it remains thin, but the inner walls and the radial walls, they become thick. And they become thick because of the presence of fibrous thickenings. And those fibrous thickenings, they are made up of alpha cellulose fibers. So, outer wall, it remains thin because of the absence of fibrous thickening, but on inner walls and on radial walls, they become thick in nature due to the presence of fibrous thickening, which are composed of alpha cellulose. So, this is the outer layer, that is the epidermis. And next to epidermis is endothecium. And this endothecium have three walls: outer wall, inner wall, and radial wall. Both epidermis as well as endothecium, they are single celled thick, as we have already discussed. This is the endothecium. Now we are considering that if this is the outer wall, then this is thin because of absence of fibrous thickening. But this radial and inner walls, they are having this alpha cellulose fiber thickening, which are known as fibrous thickenings. So, radial walls and inner walls have this thickenings. So, radial walls and inner walls, they are having the fibrous thickenings. This is the outer wall. and this is inner wall having fibrous thickenings, and this is a radial wall of endothecium. Both of these are having fibrous thickenings. Both are having fibrous thickenings. In case of radial walls, there are certain bands which are known as callous bands. There are certain bands which are known as callous bands, and these callous bands, they are present only on the radial walls, only on the radial walls. So these radial walls, they are also have callous bands. They are also having the callous bands. Now at some places, these callous bands as well as the fibrous thickenings, they are absent, and those regions where these callous bands and fibrous thickenings, they are absent on the endothecium wall, they are termed as stomium regions. So this part is known as the stomium region. This is the stomium part or stomium region. The dehiscence of the anther takes place from these stomium regions only, where these fibrous thickenings as well as callous bands are absent on these endothecium cells. So after discussing about the two outer layers, that is epidermis and endothecium, the third layer which is present inside the endothecium, the third layer is known as the middle layer. That middle layer is parenchymatous in nature, means they are composed of parenchymatous cells. Since we know that parenchyma mainly helps in the storage of food, so the main function of this middle layer is also the food storage. It is also 1 to 3 cells thick. As we know that epidermis and endothecium both are single cell thick, but middle layer it is 1 to 3 cell thick in nature. It is parenchymatous, so it helps in the storage of food. It is short-lived, means it is ephemeral in nature. After some times, the food is being absorbed by the tapetum layer, so this middle layer gets degenerate, so it is ephemeral. Ephemeral means short nature or short-lived. So in case of the mature anther, these middle layers, they are absent in those anthers. In NCERT, one line is given that makes confusion or create confusion among the students. That is, the outer 3 wall layers perform the function of protection and help in dehiscence of anther. Since we know that at maturity or in the mature anther, the middle layer gets degenerate, but till the middle layer is present, the outer 3 layers, that is epidermis, endothecium and middle layer, perform the function of protection. Although the exclusive function of protection is performed by the epidermis, but these 2 layers also somewhat provide protection. But as the anther get mature, this middle layer degenerates. So ultimately we can say that outer 3 layers, they helps in the protection, and we said that it also helps in the dehiscence of anther. This dehiscence of anther, it is being carried out or it is being performed only and only by the wall of endothecium. That's why this line is given that outer 3 wall layers perform the function of protection and help in dehiscence of anther. So outer 3 layers, they helps in protection, but the endothecium only helps in the dehiscence of anther. Since only 2 layers is present during maturity, that is epidermis and endothecium, so epidermis is involved in the protection and endothecium is involved in the dehiscence of anther. Now the 4th layer is the tapetum layer, which is known as the nutritive layer also, because it absorbs the nutrition from the middle layer and provide to the developing microspores. That's why it is considered as the nutritive layer. this layer is present inside the endothecium, so it is considered as the innermost wall layer. It is also single celled thick or single layered thick. It possesses the dense cytoplasm and it generally have more than one nucleus, means it is multinucleated in condition. Initially it is diploid in nature, but later on it becomes polyploid. This condition is transformed from diploid to polyploid. It is because of endomitosis, and it becomes multinucleated. It is because of free nuclear division. It becomes multinucleated. It is because of these free nuclear divisions. Continuous karyokinesis takes place without the cytokinesis, that is the free nuclear division. So the tapetum cells, they are initially diploid, which later on becomes polyploid due to endomitosis, and they become multinucleated in the later stages due to free nuclear division. Tapetum, it absorbs the food from the middle layers and provides nutrition to the microspore mother cells, or you can say the developing microspores. The tapetum also disappears when the anther gets mature. Once the anther gets mature, this tapetum also degenerates. So in mature anther, only two wall layers are present, that is epidermis and the endothecium. Middle layer and tapetum gets degenerated during maturity. But before the degeneration of the tapetum cells, they form certain special granules, and these special granules, they are termed as pro-Ubish bodies. So these pro-Ubish bodies are formed inside the cytoplasm of the tapetum cells. These pro-Ubish bodies, when surrounded by the sporopollenin, which is a polymer of carotenoid, these pro-Ubish bodies gets transformed into another bodies which are termed as Ubish bodies or orbicules. So before degeneration of the tapetum cells, there are certain special kind of granules are formed which are known as pro-Ubish bodies, and these pro-Ubish bodies, they are surrounded by the sporopollenin, which is being secreted by the tapetum cells, and when these sporopollenin surrounded these pro-Ubish bodies, they form the Ubish bodies. At last, once the Ubish bodies are formed, the tapetum cells gets degenerate, and these Ubish bodies get released into the pollen sacs. So Ubish bodies, which are mainly composed of the sporopollenin, they are involved or they participate in the formation of exine around the microspores, and when these microspores becomes thick walled, they are termed as pollen grains. So I think you understand this terminologies, that is what are pro-Ubish bodies, what are Ubish bodies. I can again explain whole thing by one of the simple diagram, that if suppose this is the tapetum layer and these are the special type of granules formed which are known as pro-Ubish bodies. So this is the tapetum, and these are the some special type of granules known as pro-Ubish bodies. When the tapetum cells, they secretes the sporopollenin, then those sporopollenin, they surrounds these pro-Ubish bodies. Now these pro-Ubish bodies termed as Ubish bodies. After degeneration, these Ubish bodies, they are released inside the microsporangium. Suppose this is the region, this is the microsporangium. So from the tapetum region, these Ubish bodies are being released inside the microsporangium, where these developing microspores are present. When these Ubish bodies, they are released inside the microsporangium, they formed exine around these microspores. Around these microspores, they form these Ubish bodies, they participate in the formation of exine layer. Now these microspores, they become thick walled in nature. So now they are termed as the pollen grains. Now what are the functions of tapetum? Functions of tapetum, first of all, as it is a nutritive layer, means once it absorbs the nutrition from the middle layer, it provides that nutrition to the MMC, yani microspore mother cell or pollen mother cell or developing pollens. Means it nourishes the developing microspore or developing pollen grains. Second function of tapetum layer is, or tapetum cells is, they also helps in the secretion of enzyme, and here that enzyme is Calase enzyme, and also helps in the secretion of hormone, that is particularly it helps in the secretion of indole acetic acid, that is IAA. We further discuss in the later slides that what is the function of this Calase enzyme which are being secreted by the tapetum cells. Next function is they also helps in the synthesis or secretion of sporopollenin. This sporopollenin, which is a polymer of carotenoid, it helps in the exine formation. Then tapetum cells, as we have already discussed, that they also helps in the formation of Hubich bodies, which mainly composed of this sporopollenin. Tapetum cells, they are also helps in the secretion of pollen kit substances. There are certain plants which are being pollinated by the insects. In such kind of plants, their pollen grains, they are having some extra layer outside the exine, which is known as pollen kit, and that pollen kit substances are mainly the lipids or the carotenoids, which are also being synthesized by the tapetum. So we can say that the tapetum also involved in the secretion or synthesis of pollen kit substances. Now here we discuss about the structure of microsporangium. So first of all, what is microsporangium? This sporangium terms denotes that structure that bears the spores, and here the microspores are formed inside the sporangium, so this is termed as microsporangium inside anther. If we are taking the transverse section of a typical microsporangium, then it appears circular in outline. When the anther is young, then a group of compactly arranged homogeneous cells are present, which are termed as sporogenous tissue, which occupies the centermost position of the each microsporangium. Since this microsporangium that bears the microspores, and later these microspores get transformed into pollen grains, so we can say that these microsporangium are transformed into pollen sac. So these pollen sacs, they are just equivalent to that of microsporangium. So 4 pollen sacs are present in the anther. Inside these pollen sac, microspores, or you can say pollen grains, they are present or they are formed by the meiotic or reductional division of microspore mother cells or pollen mother cells. As each cell of sporogenous tissue which is present inside the microsporangium is capable of giving rise to microspore tetrad, so we consider that each one of the sporogenous tissue is a potential PMC or a potential MMC, or you can say potential pollen mother cell or a potential microspore mother cell. This is the transverse section showing the microsporangium, which also indicates the 4 wall layers of the anther that we have already discussed, starting from outer to inner. Outermost one is the epidermis, then inside the epidermis is endothecium, then middle layers, and then the tapetum. We have already discussed that this endothecium, whose outer wall is thin in nature, inner wall and radial walls, they are having the fibrous thickenings of alpha cellulose. Inside this is the middle layer, and this is the innermost wall layer of the anther, that is known as the tapetum. And this region is which is nearly circular in outline. See this region, this is nearly circular in outline just inside the tapetum region, and this shows the microsporangium region, which is composed of sporogenous tissues which differentiate or on maturity get transformed into microspore mother cells. So each of the sporogenous tissue, it has a potential of being a PMC or MMC, which later on undergo meiotic division to form the microspores or pollen grains. Now here we are starting about the development of anther and microsporogenesis. The anther appears as a outgrowth like structure. Initially, the anther, it is just like a outgrowth of meristematic cells. So it is a outgrowth like structure in the initial stage, which shows spherical or oval shaped. At this stage, a mass of meristematic cells are present, which are surrounded by a single cell thick layer, which is known as epidermis. So it is the mass of meristematic cells which is surrounded by a single cell thick outer layer, which is known as epidermis. Now first of all, what will happen, that in the middle region vascular tissues are formed, which is known as procambium or procambial strand. So first of all, these vascular tissues are formed which are present in the middle region. Simultaneously, a group of cells which are located just below the epidermis in the vertical rows of hypodermal region at the four corners, they become large in size, and these cells are known as archisporial cells. So those cells which are just besides or just beneath the epidermal cells in the hypodermal region, those cells, they become enlarged in size, and such cells are known as archisporial cells. Now these archisporial cells, they divide periclinally. So what do you mean by periclinal division? Any division which is parallel to that of tangential plane, that is known as periclinal division. So these archisporial cells, they undergo periclinal division to form the primary parietal cells and primary sporogenous cells. These primary parietal cells, they are formed towards the epidermis, and these primary sporogenous cells, they are formed towards the center. This is the explanatory explanation of the development of anther. That is, first of all, this is the outgrowth of meristematic cells. This is the TS of anther showing the outgrowth of meristematic cells surrounded by the epidermis. Now first of all, in the middle region, this procambial strand is formed. This is the procambial strand, or you can say the vascular tissues, they are formed in the middle region. Just below this epidermis, there are certain cells in the hypodermal region. At the four corners, some of these cells, they get enlarged in size, and now these enlarged cells of the hypodermal region, they are termed as archisporial cell. These archisporial cell, they now undergo periclinal division. When these archisporial cells divide periclinally, just like this, when they divide like this periclinally, if this is the division, then one cell is formed towards the epidermis and another cell is formed towards the center. So the cells which are formed towards the epidermis, they are the primary parietal cells, and those cells which are formed towards the center, such cells are termed as primary sporogenous cells. So when archisporial cells undergo periclinal division, two cells are formed, one is outer and another one is towards the inner side. Outer side cells are formed, they are the primary parietal cells, and the cells which are formed towards the inner side, they are the primary sporogenous cells. These primary parietal cells, they undergo periclinal as well as anticlinal division. They undergo periclinal as well as anticlinal division. As we know that periclinal divisions are those which are parallel to the tangential plane, but anticlinal divisions are those which are perpendicular to the tangential plane. So these primary parietal cells, they undergo both type of division, that is periclinal and anticlinal, and this leads to the formation of three wall layers, that is endothecium, middle layer and tapetum. This endothecium, it is one cell thick, it is 1 cell thick, it is 1 to 3 cell thick, and this tapetum is also one-celled thick in nature. Now this primary sporogenous cell, it undergo mitotic divisions. So when they undergo mitosis, they forms the sporogenous tissue. This sporogenous tissue at maturity or when they undergo differentiation, they form the microspore mother cell or pollen mother cell. These microspore mother cells, they undergo meiosis to form the microspore tetrad. They form the microspore tetrad. All these 4 microspores, they are surrounded by the callose. They are surrounded by the callose. That's why they are formed in tetrad-like structure. These linings of callose, they are being degraded by the enzyme callase, which are secreted by the tapetum cells. And all these 4 microspores in a tetrad form, they get released or they become free. These are the 4 microspores when callase enzyme degrade this callose layer. This is the callose. So this callose is being degraded by this callase enzyme, and all these 4 microspores, they get free. When these microspores, when they get thickened in nature, when they become thick walled, now they are termed as pollen grains. They become thick walled, so these thick walled microspores, they are termed as pollen grains. So we have already discussed in the diagrammatic explanation, those lines are again here, that when the primary parietal cells, when they undergo periclinal and anticlinal division, they form a series of 3 to 5 layers, making the walls of the anther, that is endothecium, middle layers and tapetum. That means these primary parietal cells, they are involved in the formation of anther walls, that is endothecium, middle layer and tapetum. The cells which are formed towards the inner side, they are termed as primary sporogenous cells. They divide mitotically, they undergo mitotic divisions, and they form the sporogenous cells or sporogenous tissue. When these sporogenous cells or sporogenous tissue differentiate, they give rise to microspore mother cells. And when these microspore mother cells undergo meiosis, they form 4 haploid microspore or the pollen grains. So this process of the formation of microspore from pollen mother cells through meiosis is termed as microsporogenesis. This is termed as microsporogenesis. This is the microspore mother cell having a diploid nucleus. When it undergo meiosis, that is the first step of meiosis, that is meiosis 1. When it undergo meiosis 1, it forms the 2 haploid nuclei. Then further, when it undergo meiosis 2, it again forms 2 more haploid nuclei. To total 4 haploid nuclei are formed after the completion of meiosis. So when MMC or microspore mother cell undergo complete meiosis, it gives rise to microspore tetrad. This forms the microspore tetrad, and all these 4 microspores in a tetrad, they are being free when the callase enzyme degraded the callose wall. So in this lecture, under the heading of pre-fertilization events, we had discussed about the male reproductive world, that is androecium, whose basic unit is stamen. In this lecture, we also discussed about the structure and the development of anther, that how the anther gets developed from an outgrowth like structure which is a meristematic cells. And also we discussed about the walls of the anther, that is about the 4 walls of anther, that is epidermis, endothelium, middle layers as well as tapetum. And finally, here we discuss about the microsporogenesis, that is the formation of microspores from microspore mother cells, that how the sporogenous tissue when gets mature into microspore mother cell, and that microspore mother cell undergo meiotic divisions to form the microspores, and these microspores when get thickened they are known as pollen grains. Thank you.